Hello, hi everyone. Welcome to this M2D Tool Talk. Our speaker today is Zuobai Zhang, and he will be presenting his recent paper titled Protein Representation Learning by Geometric Structure Pretraining. Zuobai is a PhD candidate at Mila, advised by Professor Zhang. His research interests include the design of graph neural network and their application in drug discovery, especially protein structure representation learning. And he also led the development of open source libraries such as Touch Protein, to facilitate the usage of machine learning model in protein science. Thank you so much, Zuobai, for accepting to present your work to us today. And yeah, without further ado, the floor is yours.
Okay, so thanks for Prudential's introduction and thanks for everyone's coming. And hello everyone, I'm Zuobai Zhang. I'm currently a second-year PhD student from Mila, advised by Professor Zhang. And my research interests mainly include protein representation learning, and today I'll present our recent work, Protein Representation Learning by Geometric Structure Pretraining. This work was done by Minghao Jin and me from Mila, Erin from University of Cambridge, Vijay Ahuridi and Peel from IBM Research. And this work is published at this year's Academia. And feel free to ask questions during the presentation. You can ask questions in the chat and I will stop at the end of each part to answer some questions. Okay, so let's get started. So first, in this work, we will focus on protein representation learning. Proteins play fundamental roles in our life. For example, they can serve as an enzyme to catalyze some chemical reactions. Also, they can transmit some signals in the cells. Besides, they can also serve as antibody to bind with some antigens and serve as a self-defense mechanism in our body. Therefore, protein representation learning is an interesting and important problem in both machine learning and biology community. We want to learn some useful representations for proteins, and then we can do some downstream tasks. So let's first start with some basic components of proteins. The basic components of proteins are called amino acids, also known as residues. So each amino acid will consist of an amino group, a carboxylic acid group, an alpha carbon atom, and a cytosine group R, identifying the type of this residue. And with these basic components, residues, we can a protein will be formed by one chain or multiple chains of residues. And the adjacent residues in the chain will form a peptide bond through dehydration reaction as shown in the figure. So the carboxylic acid amino group with the amino group will have some dehydration reaction, and then they will form a peptide bond. And in this way, we can connect consecutive residues to form a protein sequence. And here, we use one character to denote one type of residues. And typically, we assume that there are around 20 types of standard residues in the nature. And we use one character to denote one types of residues. And in this way, we can have protein sequence. And protein sequence contains most essential information about the protein. So given protein sequence, what can we extract from this information? So first, we know that the protein sequence directly determines its 3D structures, also known as conformation. And here by 3D structures, we mean the 3D coordinates of all the atoms in the structure, in the protein. And this has been recently well solved by recent protocols like AlphaFold2 and ESMFold2. We can get accurate predicted protein structures from the protein sequence efficiently. And protein structures are known to determine their functions directly. For example, given a protein structure, we will know whether a protein can bind to an antigen, can bind to another protein or some small molecules, and we can know that whether it can catalyze some chemical reactions, or we can know some of its properties like thermal stability or aggregation. So all of these functions of protein functions or properties are very important for protein design, because the final objective of our protein research is to design some proteins with desired functions properties. So in this work, we will focus on protein function prediction, and we will see how to use structure to help protein function prediction. So previously, obtaining protein structures is very expensive. We need some experimental approach for some wet lab experiments to get the experimental structures. So previously, most machine learning methods are directly based on the protein sequence, because protein sequence contains most essential information about protein structures. And these methods can be categorized into two kinds. So the first kind is supervised learning. So basically, we can design some protein sequence encoder, for example, convolutional neural network, STM, or transformer. And then we use the labeled protein function data to train the protein sequence encoder, and we do the supervised learning test on some unseen data. However, protein function label are very sparse, so we cannot get a very good model if we only use the protein function data. So there is another kind of works called protein language models. They falls into a pretraining and fine-tuning framework. So basically, these models are mostly based on transformers, and they want to utilize the unlabeled protein sequence data. So we have billions of protein sequences available in the nature in our database, but we do not have its function label. But we can still use them with some pre-training mechanism, for example, mask language modeling. So these methods are referred as the protein language models. Two typical examples are ProtBERT and ASM1B. And they will pre-train the model on some large-scale sequence database and fine-tune it on some downstream function prediction data. However, at 2021, a game changer come out. So that is the AlphaFold 2. And inspired by recent breakthroughs in the protein structure prediction protocol, we can now predict the protein structures from its sequence accurately and efficiently. So now we have much more available protein structural information. So in this work, we will focus on the specific problem is that, can we use this protein structural information to help our protein function prediction? Because we know protein structures are the most direct determinant of protein functions. So it contains much more valuable information in protein structures. In this way, we are expected to get a good protein function predictor. So that's what we want to do in this work. So specifically in this work, we will focus on structure-based protein representation learning. So given a protein structure, we want to learn a good representation. So we design our methods following the previous two kinds of methods in sequence encoders. So first, we focus on supervised learning setting. So we design a protein structure encoder based on graph neural networks and protein structure graph. And this encoder is called GearNet-Edge. And after we get a good protein structure encoder, we want to utilize the large-scale unlabeled structure data. So we want to design some self-supervised pre-training methods based on the protein structures. And so here we propose a multiview contrast pre-training methods based on the contrastive learning. And also we propose 4 additional self-prediction pre-training baselines. So that's the motivation and the main contribution of our work. And any questions so far? Okay, so if no questions, maybe we can quickly move to the first part. So next, I will present our GearNet. So this is a simple yet effective protein structure encoder. It is based on the protein structure graph and some typical graph neural networks. Okay. So before we start to discuss some protein structure encoder, maybe we can quickly review how we design the protein sequence encoder, and then we can discuss how to generalize the protein sequence encoder to protein graphs. So let's use the simplest example, the convolutional neural networks. So start with the convolutional neural network. Let's think about how can we apply the convolution on a protein sequence. So here we have a protein sequence. We have one character to denote one type of residue. And we want to apply a convolutional layer on this sequence and to get an equal length output. So what we typically do is that we can add zero padding at both ends of this sequence, and then we can define a convolutional layer iterating over all the protein sequence. So that's the high-level idea of convolutional neural network. And what should we note here is that, so first, the convolutional layer is shared among all the different positions. So we use one convolutional neural network iterating all over the whole protein sequence. So that's the first point. And if you focus on one specific convolution layer, you can see that we will assign different weights according to the relative positions of input and output. For example, you can see we will, so the first position, the relative position between the first point and the output is minus 1. So that's the sequence distance. And the second position is 0. The third position is 1. So that's how we define the sequence distance. And for positions with different relative sequence distance, we will assign different weights. And that's how we define a convolutional neural network, a convolutional layer. And by stacking multiple convolution layers, we can get our convolutional neural networks. So that's how we define the convolutional neural network on protein sequence. And now let's think about how to generalize this to protein structures. So the essential problem is that we want to think about how to generalize the convolutional neural network from protein sequence to protein structure graph. Okay, so basically that's how we deal with the protein sequence information. And here, now we can have both protein sequence and protein structural information. And now we want to represent the protein sequential and structural information in a multi-relational graph. So we define a graph based on this information. So first, given a protein structure, we will extract the alpha carbon of each residue. And then we treat this atom as the nodes in our multi-relational graph. So in this way, we can construct a residue level graph where the number of nodes in our graph is equal to the number of residues in the proteins. Yeah, so that's how we consider the nodes in the graph. And next, we want to discuss how can we construct the edges between nodes. Typically, in previous works, if we want to use protein structures, we only consider some spatial edges. For example, if the spatial distance between two nodes are below some threshold, we will add an edge between them. But this is not enough because we also want to include the protein sequential information. So how can we include both sequential and structural information into one graph? So the answer is multi-relational graph. So first, to encode the sequential information, we can connect each residue to its next residue. So we add an edge between them. And besides, we will add an edge back to them. So we link each residue to its previous residue. And these two edges are treated as different relations. I will explain later. And so in practice, we can use different sequential distances. We can also link some edge from 1 to 3, 2 to 4. So we can use different sequential distance threshold. So that's how we include the protein sequential information in our multi-relational graph. And besides, we can also consider some protein structural information by consider the 3D structures of each of the protein. So we consider the 3D coordinates of each atoms, and if two atoms are closed in the Euclidean space, we will add a radius edge between them. And we also consider the k nearest neighbor edge. So for each node, we will connect it with the k nearest neighbor in the space. And in this way, we can construct a multi-relational graph. So why should we treat these relations as different? Treat these edges as different relations according to the ways we construct this edge. So remember, in the convolutional neural network, we will assign different weights according to the relative positions between the input and output. So if the weight is decided by the relative sequential distance in the convolutional layers. And here we can use the similar idea. So the type of each edge indicates the relationship between the two nodes. So we can see if there is a yellow edge between two nodes, we know that the first node is the previous residue of the second node. And if there is a red edge between two nodes, we can know that there is some spatial connection between them. So that's why we want to define a multi-relational graph. And next, after we get our multi-relational graph, the idea is simple. Oh, I see. So there is a question.
Yeah, there's a few questions in the chat relating to the previous slide.
Okay, so radius graphs are also directional, but in practice, we know that if there is a radius graph pointing from 3 to 4, there will also be an edge pointing from 4 to 3, right? So in practice, we will add two edges between 3 and 4 with reverse directions. And what's the rationale behind using both radius edge and the KN edge? Seems okay. So that's a very good question, and I want to leave this until the end of our presentation. So, so considering the time limits, I may not answer this question now, but I will answer it at the end of this presentation. Okay, so maybe we can quickly go through and.
Maybe before we move, Husseini has a question.
So I see Archean radius directional graph like SimCLR and protein structures and we map directly. It's true that knowing structures. Okay, so actually we think protein sequence can contain most useful information about the protein functions, but in practice, it will be better to know the protein structures because so protein structures are the most direct determinant of protein functions. If you want to know whether a protein can bind to an antigen, you will derive the binding affinity based on protein structures, right? And it's like we provide more input to our model, and in this way we can learn better representations about the protein function. Yeah, and maybe I'll leave some questions to the end of this presentation, and I will go ahead about this part. So after we construct the multi-relational graph, now we can perform the relational message passing on the graphs. So it's very similar to the graph convolutional neural network. The only difference here is that we will assign different weights to different relations. So for example, in this graph, if we look at the node 4, we take all its neighbors in the graphs, and we can see there are 4 kinds of relations in the neighborhood. And here for each kind of relation, we will assign a different weight matrix, different convolutional kernels. And by performing relational message passing, we mean that we use different convolutional kernel for different relations. And after that, we will get the message functions on the edge and do some aggregation to get the final node representation. So that's how we define one relational message passing layer. And by stacking multiple relational message passing layer, we can get our geometry-aware relational graph neural network, which is also called GearNet. And in this way, we can generalize the convolutional neural networks to graphs. Yeah, so that's the basic definition of our GearNet model. So previously we only focused on node level message passing, but node level message passing is not expressive enough. Consider in AlphaFold 2. In AlphaFold 2, they want to learn the pair representations between all pairs of residues in the graph in the protein. And they will use a triangle update to learn the residue representation, to learn the pair representations. So they consider all triplets of residues and use triangle updates to learn the pair representations. And in our work, because we only consider a sparse graph, so in our work, we will consider a sparse version of a triangle update. So instead of learning the representations between all pairs of residues, we only consider the edges in representations in our graph. And so instead of performing the triangle update between all pairs, between a fully connected graphs, we will build a graph between the edges. So in the new graph we call line graph. In the line graph, we will treat the edges in the original graph as nodes, and we will connect edges with a common end node. For example, here, if we consider the edge from node 4 to node 7, and then we can build a new line graph on the edges in the original graph. And here, we can see we will construct its neighbors by linking all the edges pointing to node 4. So here we add, so there is two edges with different relations pointing from 2 to 4 in the original graph, and in the new constructed line graph, we will add the edge from the edge 2, 4 to edge 4, 7. And similarly, we can construct the neighborhoods of the edge. And here we will also consider a multi-relational graph. And how we decide the relations, we will use the angles between the two edges to define the relations. For example, the relation of between 2, 4 to 4, 3 is defined by the angle between 2, 4, 3. 2, 4, 7. So it's defined by the angles formed by the 3 residues. In this way, we can include the angular information by considering atom relation in the line graph. So after that, after we construct an edge graph, now we can perform the relational message passing again. So the only difference here is that we do not operate on the original graph. We operate the line graph, and each node on the line graph corresponds to an edge in the original graph. And by performing the relational message passing, we can include all the angular information and to learn the edge representation. So that is the edge message passing layer. And by aggregating the edge information into the node representations, we can enhance our GearNet with the edge message passing. And this model is called GearNet-Edge. Yeah, so that's the high level idea of our encoder. And basically we include the relational information and relational message passing and edge message passing. And in the experiment, we focus on 2 function prediction tasks. The first one is enzyme combination. Basically, we want to classify whether a protein can catalyze a chemical reaction. And the other one is a gene ontology terms classification. So basically, we want to distinguish whether a protein can perform a molecule function, a biological process, and what's the cellular location that the process will happen. And both of these tasks are multiple binary classification tasks, and we define a metric called Fmax based on the F1 score. So this metric is the larger, the better. And now we can show you the results of a method. And here you can see, we compare our GearNet methods with some sequence-based representation learning methods, including four sequence-based methods. And as you can see, the GearNet results are shown in orange, and it is much better than sequence-based results. So we can see that structural information can provide very valuable information about function prediction. And by further augmenting our model with edge message parsing, we can see that GearNet-Edge can outperform GearNet by a large margin. So we can see GearNet-Edge is the best model among all the baselines without pretraining. Yeah, so that's for the supervised learning part. And I think since the time is limited, and I think there won't be enough time for me to stop and answer questions during the presentation, you can ask questions in the chat and I will answer in the final Q&A session. So let's go ahead. So after discussing the supervised learning part, let's move on to the pre-training part. So first, why do we want to do the pre-training? Because we know labeled protein function data are very sparse, and but we now have massive unlabeled protein data, both the protein sequences and the protein structures. For this data, we do not have its protein function label, but we know it, and we want to utilize this unlabeled protein data. And previous methods mainly focus on use the unlabeled protein sequence data, and in this work, we want to use the unlabeled protein structure data. And now we already have a good structure encoder, which is our GearNet-Edge. And we also have a large unlabeled pre-training data set, either the protein data bank or our outperform database. And now we only need, what we only need is a good structure-based pre-training algorithm. And that's the part we want to focus on now. Yeah. So let's quickly review the previous geometric pre-training methods. So typical pre-training algorithms can be classified into two categories. So the first one is contrastive learning. So basically, given a training example, we will first construct a correlated view by perturbing it. And then we feed the two views into the same model to get its representation. And since we know the two views are correlated, their representations are expected to be similar. So we will minimize the distance-based loss, or just to maximize the similarity between the two representations. So that's the high-level idea of contrastive learning, to maximize the similarities between correlated views. And the other kinds of pre-training methods is called self-prediction methods. Basically, given a training example, we will still perturb it with some mask. And after getting the perturbed example, we will feed it into our model and to use the model representations to reconstruct our training example. A typical example is the mask language modeling used in BERT. Basically, we first mask out some word in our sentence and then feed the sentence into our transformer model, use the output embeddings to predict back our word. So that's the self-prediction method. And the only question here is that how can we generalize these methods to protein structures? And now we can answer these questions one by one. And let's first focus on the contrastive learning part. So one typical method in contrastive learning is called SimCLR. And this is one famous work in computer vision community. So basically, given the image, we can generate two different views by applying some augmentation of view functions. And after that, we will feed these views into a convolutional neural network or some encoders to get representations, and then feed into an MLP to get a latent representation. And then we will align or attract the representations from the same image, and then minimize the similarity between representations from different image. So that's the high-level idea of the contrastive learning. And they will apply info ACE loss. Here, Z refers to the latent representation. And we will maximize the similarity, cosine similarity between the representations from the same image and minimize the similarity between the views from different image. So the only question is, how can we generalize these contrastive learning methods to protein structures? And these questions can be transformed to another question, is that how to design correlated views for protein structures. And after we get correlated views, we can apply the same framework to use influence loss to maximize the similarity between representations. And in this work, we propose a method called multiview contrastive learning to solve this question. So basically, given a protein structure, we can still construct its protein multi-relational graph as we have discussed before. And after that, we can generate two correlated views by randomly applying some view functions or augmentation functions. And here we consider 4 different view functions, which are subsequence cropping. So we randomly select a consecutive subsequence for the protein, and a subspace cropping. So first we randomly select a residue at the center, and then consider radius in the Euclidean space to select all the residues in the ball. And also a random edge masking operation to add noise to the relational graph. And finally, identity function. And by randomly choosing one of the 4 correlated view functions, we can generate 2 correlated views, Gx and Gy. And after that, we will apply an influence loss to align the representations from the same proteins and minimize the similarities between the views from different proteins. So that's the high-level idea of multi-view contrastive learning. So it's essentially very similar to the SimCLR, but here we propose several view functions defined specifically for proteins. Yeah, and next we will discuss how to design self-prediction baselines. So basically, the idea is also very simple. So if you think about the mask language model, so basically we want to mask out some residue types and then use the representations to predict back. And to utilize the structural information, we also consider other self-prediction baselines. For example, we will mask out the edges between 1 and 3, and then use the representations to predict the spatial distance between these 2 residues. And also we can consider the angle prediction. We will mask all the edges between 1, 2, 4, and then we can use the representations of these 3 residues to predict the angles formed by them. And also we can consider the dihedral predictions. So we consider 4 residues in the graph, for example, 1, 2, 4, 7, and then we mask all the edges between them and use the representations to predict the dihedral prediction, dihedrals formed by these 4 residues. So in this way, we can enforce our representations to encode the spatial or structural information about the proteins. And in this way, we can make sure that our representations can encode some useful structural information. Yeah, so that's the high-level ideas of our multi-view contrast methods and self-prediction baselines. And next, we will do some experiments by pre-training our GearNet-Edge encoder with this method. So we use the AlphaFold database version 1 and version 2 as our pre-training database. So we use this database because this is the largest database at the time we wrote the paper. And currently we are working on pre-training our encoders on the entire AlphaFold database to use the hundreds of millions of structures. And for AlphaFold database version 1 and version 2, we only have around 800,000 protein structures. So we still use the EC and Geo prediction task as the evaluation. We compare our methods, GearNet-Edge, and we also compare our methods with some sequence-based pretraining methods. And among them, the best one is ESM1B. These sequence-based pretraining methods are typically pretrained on millions or even billions of protein sequence and takes much longer time for pretraining. And in our method, we only pretrain our model on fewer than 1 million protein structures. So we use much less pretraining data. So the first thing we can observe is that by using structural pretraining algorithms, we can achieve significant improvement compared with the GearNet-Edge without pretraining. So if you look at the numbers, orange numbers and the green numbers, we can achieve significant improvements. And compared with sequence-based methods, we find that we can achieve comparable or even better performance with much less pretraining data. So that's the main advantage of structure-based representation learning. So in this way, we can highlight the importance of using structures for protein function prediction. So one question you may ask is that why do we use the predicted protein structures in AlphaFold database? Why don't we just use the experimental determined structures in protein data bank? So here we compare the pretraining performance on the different pretraining datasets. So we pretraining our, so for all the 5 structure-based pretraining baselines, we pretrain our model on the four kinds of database. So for different versions of AlphaFold database and PDB. And we can see that on EC, so there is no clear advantage of using use PDB as our pretraining dataset. So you can see sometimes using PDB is better, but sometimes using AlphaFold database is better. So so to conclude, in this way, we want to use the use a much larger pretraining database instead of only use the more accurate database. So I think it's reasonable to use AlphaFold database as, for pretraining. And another question you may ask is that, so after you're designing some complicated structure-based methods, it seems that you haven't achieved some improvement beyond the protein language models. So I think the main reason is that we haven't scaled our model to the whole AlphaFold database. And to solve this question, we can think about how to combine the structure-based methods with the protein language models. And in our recent works, so this is a workshop paper in also in this year's academia. So we propose to combine our structure-based methods with the protein language model. So the idea is also very simple. So we first feed the protein sequence into the ESM or some other protein language models, and then use the protein representations as use the residual representations as the node features in our GearNet model. And by just feeding the ESM1B's output into GearNet, we can get a new model. We call this ESM GearNet. We will see that this model is very simple, but very, very effective. It can achieve much better performance, even without pretraining. And also we propose that we can combine the sequence-based and structure-based pretraining by just use the multiview contrast method on the ESM GearNet encoder. So the idea is still very similar. So given a protein structure, we will generate correlated views, and now we feed it into the ESM GearNet to get representations and align them in the data space. And in this way, we can combine the sequence-based pretraining in ESM and structure-based pretraining. And now we show the results. So here we show the results for GearNet, GearNet-Edge, and ESM1B, and also GearNet-Edge pre-trained with multiview contrast. So we can see that after augmenting ESM with GearNet, we can achieve better performance than simply use ESM1B. So you can see the performance improvement is quite large. It's about 3%, and by further pre-training our ESM GearNet with multiview contrastive learning. So in this way, we can combine both structure-based and sequence-based pre-training methods. We can further achieve some improvements. So in this way, we can use structure-based methods to enhance the protein language models. And besides these works, we also developed an open source library called Touch Proteins, because we find doing machine learning on proteins needs some sophisticated chemical or biological features processing, and we need to handle different modalities of the protein, for example, protein sequence or protein structures. So we developed this Touch Protein library, and this Touch Protein library will provide unified data structures for representing proteins, and also flexible machine learning building blocks. And we provide extensive benchmarks and pre-trained model in our library. And to conclude that in this paper, we studied the structure-based representation learning. We find structure-based representation learning can help function prediction. And we propose a simple yet effective protein structure called encoder called GearNet. So essentially, we use relational message passing and edge message passing. And besides, we propose one contrastive learning-based geometric pre-training algorithm, and also 4 self-prediction pre-training baselines. We find that these methods can achieve good performance on function prediction tasks. So one thing I want to emphasize is that you can see most of these, most components in our methods are inspired by previous works. But I think it's still important to generalize these works on protein structures. And actually in this work, we have shown that structure-based representation learning is really helpful and important. And for some future works, there are something I'm working on, and I think it's promising to work on. So the first part is how to combine the protein sequence and the structure pre-training. And I think it's very beneficial because sequence data, although it contains less information, less direct information than protein structures, but we have much more protein sequence data. So it's important to combine both kinds of pre-training methods. So we have 2 recent papers talking about that. So the first one I have introduced is published in this year's SimCLR MLDD workshop. And another one is called SimDiff. Basically, we combine the sequence and structure pre-training with some diffusion methods. And so that's the first direction. And the second direction is what I'm working on is to try to pre-train our structure-based encoder on the entire AlphaFold database. And after scaling our models to such a larger scale, we can see whether structure-based method can really achieve significant improvement over ESM1B. And besides, we also want to consider some downstream tasks, for example, protein-protein interactions, protein engineering tasks, and protein design. In all of these tasks, protein structures play very important role, and we want to see whether structure-based pre-training can help these downstream tasks. And maybe that's all from my presentation, and thanks everyone, and feel free to ask questions now. And yeah, maybe I can start with some questions in the chat now. Okay, so let's first start with Remy Liu's question. So he asked, what's the rationale behind using both radius edge and KN edge? This seems to be correlated to some extent, right? Okay, so I have a slide to explain that. So in practice, when doing pre-training, we find that our AlphaFold database, so basically to encode the spatial information, we use the radius KN edge, and we find that so if you use a radius edge, you will have more. There will be more edges in the dense region. If such a region contain more residues, you will have more edges. And but on different proteins, on even different species, you may need to tune the radius. Otherwise, there will be some residues without any spatial edges. So there will be some residues at the end of the protein sequence, and they may not have some spatial interactions with other residues. And as you can see here, we show the average node degree on the AlphaFold database. And you can see if you only use the radius edge, which marked by in red, so there will be many nodes with degrees fewer than 5, maybe only 2 or 3. And this is not good for the message passing on it's especially for the constructing the node graphs, because you may need some you don't want a graph to be too sparse, because it will limit the expressive power of your graph neural networks. And by adding GearNet-Edge, you can guarantee that there is a lower bound for your for the node degree. Yeah, and so that's the answer for the video. And next question is, do you see the full or subset of geo terms? If it's the subset, what are some mean filter criteria? Okay, so okay, let me see. So for geo terms, so the geo dataset is directly adapted from the deep FRI. Sorry, may not showing here in this page, but a geo dataset is taken from the deep FRI benchmarks. And I think they only use a subset of geo terms, and they do some filtering on the protein sequences. So basically, they first do some sequence clustering, and then set a split threshold for the dataset split cutoff. Yeah. And so for the next question, Natalie Sennon's question: in subspace cropping, could you be cropping a part of protein sequence that is related to the central node when the protein is folding? Yeah. So here for the subspace cropping, that's the high-level idea of our subspace cropping. Because if you only use the subsequence cropping, you will not consider the protein folding information. You do not have the structural information. But by consider the subspace cropping, we can also include all the residues close in space. So we know that if 2 residues are close in sequence, they are likely to be close in the space. But there are also some residues with long sequence distance, but small spatial distance. And the subspace cropping is designed to include these residues. Okay, so next question is Aldo Zami. Working at the residue level may be enough for general protein function task, and it may not capture non-relevant 3D interactions between residue side chains. Do you think that could be a limitation of more fine-grained task looking for interesting? Yeah, that's a very good question. Actually, I'm working on that. So first, I want to say that this method it can be easily generalized to atom graphs, because the only thing you need to do is that you include, so when construct the multi-relational graph, you include all the atoms in the residues. And what you need to consider is how to construct these graphs. Because now maybe you cannot add the sequential edges between all the atoms, because it will be too dense. There will be too many edges in the atom graph. But you can consider the bound edge in each residue or something like that. So that's so by changing the way we construct the edge, we can apply our methods on the atom level. And actually, I have some experiments in our latest paper. So, sorry, in our SumDiff, in this work, we try to use the atom level graphs on the atom 3D benchmarks, and we see that it's very useful for some tasks like protein structure assessment or protein-protein interactions. So that's using atom level data is very important. And by using GearNet, we can easily generalize the GearNet-Edge to something like that. And also we have some tests on protein-protein interaction tasks in real world, in wet lab experiments. And so this work has not been published on arXiv, but will be released in the next few months. So actually, it's very interesting and doable to generalize our method on atom level. So next question is Ether's question. So have you performed structural clustering before creating training set and pre-training phase? I'm not sure the bigger set is necessarily a better one. That's also a very good question. So in the GearNet version, in this paper, we do not perform any structural clustering because here we only have less than 1 million structures. So the number of structure data is not very large compared to protein sequences. But I think it's beneficial to include some structural clustering because there are some recent works from Fold 16s. They cluster the entire AlphaFold database using their using some structure clustering methods. And I'm now trying to pre-train our methods on the clustered AlphaFold database. And I find it's beneficial to do some structure clustering. But in this work, we haven't do that. And we find that even you haven't do that, you can achieve good performance. So that's a very good question. Question of Aldo is very important for protein complex. Yes, I think I have answered this question, and we have already done some work on the atom level tasks. And the next question is Raf's question on slide 23. When you compare F1 score for EC numbers, are these all competing? Independent methods for also are some correlated, such as structure-based. Sorry, maybe I cannot understand your question. So could you explain it? So
yeah, so on the y-axis, you have these different methods like residue type prediction, distance prediction. Are you using just, like in this example, are you using just distance prediction and then calculating the F score for EC number by itself?
Yeah, right.
Okay.
So in this task, we just want to try different pre-training methods on different pre-training datasets to see whether it's a good choice to use AlphaFold database.